Hello everyone, today we will talk about neuronal electrophysiology. In this video, we're going to learn three main things. First, we'll talk about how a nerve cell stays charged when it's resting, which is called the resting membrane potential. And second, we will see what happens when it gets a small electrical signal. This is called graded potentials. And finally, we'll explore the big electrical event called the action potential, which is how neurons actually send messages. So neurons talk to each other using electricity. When a neuron is just sitting quietly, it keeps a small negative charge inside of it, it's like a battery. This is called the resting membrane potential. If something changes this charge a little, we call it a graded potential. You can see the charge of the neuron is changing. This is called graded potential. And if this change is big enough, the neuron will fire an action potential, and this is the big electrical signal that will travel all the way down to the nerve to send a message. So now we will talk about each step by step. First, we have the resting state of a neuron, which is the resting membrane potential. Imagine a neuron like a balloon filled with charged particles. When it's not doing anything, it keeps the inside more negative than the outside. This is called the resting membrane potential, and it is equal to minus 70 millivolts. We have three key things to help create this resting membrane potential as a negative charge. We have the sodium-potassium pumps, we have the leaky potassium channels, and we have the leaky sodium channels. Now we will discuss each of these factors. First, we will start with the sodium-potassium ATPase pump. So we have this pump that uses energy, which is ATP, to push sodium ions out of the cells and bring potassium ions into the cell. So every time this sodium-potassium ATPase pumps, it will send 3 sodium ions out of the cell, and it will push 2 potassium ions into the cell. So it will make the inside a bit more negative because it is pushing 3 sodium ions to the outside and entering 2 potassium ions to the inside. So the inside will be a bit more negative than the outside. Second, we have the potassium leak channels, and those are like small doors that are always open, letting potassium escape the cell, making the inside even more negative. And finally, we have the leaky sodium channels, which allow a tiny bit of sodium back into the cell, but not enough to cancel out the negative charge that is inside. So all of those factors together, the sodium-potassium ATPase pump, which is here, the leaky potassium channels, and the leaky sodium channels, all of those together will contribute to the negative resting membrane potential that is around minus 70 millivolts. Now let's move to the next step, which is graded potentials. Imagine that the resting membrane potential is now getting a little disturbed, and this is what we call graded potential. Graded potentials are a small change in the electrical charge of the neuron, which usually happens in the dendrites or cell body. And these changes can either be excitatory or inhibitory. Excitatory postsynaptic potentials that bring the neuron closer to firing and closer to action potential, or inhibitory postsynaptic potentials that push it further away from firing. So the excitatory postsynaptic potentials, these are they cause depolarization, so making the inside of the cell less negative. They are caused by the influx of positive ions like sodium or calcium through ligand-gated ion channels, so they cause influx of positive ions inside the cell, so they can make it more positive and less negative. So they will make the resting membrane potential around minus 55. So they will make it less negative and more positive. Whereas the inhibitory postsynaptic potential, they cause hyperpolarization, like here, hyperpolarization, and they are caused by the influx of negative ions like chloride, or the efflux of positive ions like potassium, also through ligand-gated channels. And the inhibitory postsynaptic potentials, they move the membrane potential further away from the threshold, so further away from being able to cause an action potential. So those are the excitatory postsynaptic potentials, which are helping the membrane to become more positive, so helping it to reach an action potential, whereas those are postsynaptic potentials, inhibitory, that are making the membrane more negative and away from being able to make an action potential. And both of those types, excitatory and inhibitory, are graded potentials. They are changes in the voltage of the membrane, but they are not yet reaching an action potential. So, as we said, we have excitatory postsynaptic potentials and inhibitory postsynaptic potentials, and those are types of graded potentials. To make it clearer for you, I'm going to show you this picture. Here we have the resting membrane potential, which is stable. It is minus 70. When this potential starts to get disturbed, this is what is called graded potentials. Those are excitatory potentials. It's trying to depolarize the membrane. And this is inhibitory, and this is excitatory, this is inhibitory, excitatory, and thereafter. So this is what is called the graded potentials. Also, there is another concept that you need to understand about graded potentials, that graded potentials don't act alone. They can add up. Actually, they should add up for reaching the threshold of action potential. So here we have the threshold. We have a certain threshold that we need to reach in order to have an action potential. So those graded potentials should add up in order to reach the threshold of an action potential. And to add up, this is called summation. We have two types of summation. We have the temporal summation and we have the spatial summation. Temporal summation means that signals keeps coming quickly from the same neuron, whereas spatial summation means that signals come from different neurons. Here we have two neurons. So we have signals coming from two different neurons at the same time. And if those excitatory signals are strong enough in both the temporal summation and the spatial summation, the neuron will send an action potential. So when the graded potentials are strong enough and they add up to reach the threshold potential, which is minus 55 millivolt, which is necessary for excitation to produce an action potential, we will have the neuron will fire and we will have an action potential. So the action potential is a big fast electrical signal that moves along the neuron like a wave. It's all or nothing. Once it starts, it will go all the way down the neuron without fading. We have three phases of an action potential. We have the depolarization, we have the repolarization, and we have the hyperpolarization. So first we have the depolarization phase. So in this picture we can see this is the resting membrane potential that we talked about. The inside of the cell is more negative. Once we will have a depolarization, which is here, the inside of the cell will become more positive in order to have a depolarization. So what happens? Something exciting happens, which is voltage gated sodium channels will open and sodium will rush inside the cell, and this will make the inside of the cell suddenly go from negative to positive up to around plus 30 millivolt. As you can see here, the membrane goes from minus 70, it depolarizes up to plus 30, so from negative to positive. And this positive charge will spread down the axon, and it will open more sodium channels. And at the end of the neuron, this will also trigger the opening of calcium channels. It is not present here in the picture, but this will happen also. Calcium channels will open, and those calcium channels will help release neurotransmitters to the next cell in order the message to be transmitted from one neuron to another. And then we have the repolarization phase, which is this phase here. What happens during repolarization? This is when the neuron tries to go back to its resting state. So sodium channels will close and potassium channels now will open. So potassium rushes out of the cell, taking positive charges with it, so which will bring the charge inside the neuron back down more negative again. So it was positive, and then during the repolarization state, potassium channels opened, potassium came out of the cell, so it turned the cell inside more negative. So this is why we can see here during the repolarization phase, the voltage of the membrane became negative. And during repolarization, when the charge inside the neuron is back to negative, this will also help to stop the neurotransmitter release at the end of the neuron. All of this that I'm saying is written here, okay? So don't worry. Everything that happens in the phases. Okay. So now we have the repolarization phase. We talked about it. After the repolarization, we will have what is called hyperpolarization. Hyperpolarization, it's here. What happens during hyperpolarization? The neuron slightly overshoots its resting state. So we can see that its resting state is -70. But during hyperpolarization, we can see that it became more negative at around -90. Why? Because potassium channels are a bit slow to close. So too much potassium leaves, making the inside even more negative than before. As we said here, we have potassium channels that opened and made potassium come out of the cell. Those channels are slow, so they are very slow, and they are also slow in closing. So they made a lot of potassium to come out of the cell. So this made the membrane even more negative than before. But what acts to maintain the resting membrane potential and to restore the same resting membrane potential? As we said, the sodium-potassium pumps, like we talked before, and the potassium leak channels and the sodium leak channels that will act to maintain the resting membrane potential. So after the hyperpolarization, those three factors will act together to maintain and to stabilize our resting membrane potential, which is at -70 millivolt. Okay, so we talked about action potentials and the three phases of action potential. And now, before we end our lecture, there is two more concepts that you need to understand. Inside the depolarization phase, we said that voltage-gated sodium channels needs to open in order for sodium to come inside the cell and make the cell more positive. So you should know that those voltage-gated sodium channels have two gates. They have what is called an activation gate, and they have the inactivation gate. So during the resting membrane potential, the activation gate is closed, but it is capable of opening, and the inactivation gate is open. But during depolarization, so from the threshold to the peak potential, from -50 to +30, so during the depolarization phase, the activation gate will open and the inactivation gate will stay open. So this will allow sodium to come inside the cell. And then during the repolarization phase, which is from +30 to -70, during this phase, what will happen to the gates of the voltage-gated sodium channel? The activation gate will stay open, but the inactivation gate will close and will not be able to open. So the inactivation gate is closed and not capable of opening. Finally, let's talk about refractory periods. After firing, a neuron needs a bit of time to recover. So we have what is called refractory periods, and those periods ensure that action potentials travel in one direction and limit their firing rates. We have two types of refractory periods. we have the absolute refractory period and the relative refractory period. The absolute refractory period is from the peak of action potential until the resting membrane potential is nearly restored. So it is between the depolarization and the repolarization phase. And during this phase, during the absolute refractory period, no matter what, we cannot have another action potential. So the neuron cannot fire again because the sodium channels are inactive and cannot open. So as we said, that during the repolarization phase, the voltage-gated sodium channels, they are closed and they cannot open no matter what. So this will limit the firing of another action. And the other type of refractory period, which is the relative refractory period, which is during the hyperpolarization phase, the neuron can fire again, but only if the signal is really strong, because the neuron is still in a more negative than usual state. So it is more negative than minus 70. It is at around minus 90. So it needs a signal that is stronger than usual. So in order it can reach the threshold, which is minus 55. So it needs a stronger than usual stimulus is required to reach the threshold and generate another action potential. We've come to the end of our lecture. You can expect questions on the factors establishing resting potentials, the difference between the excitatory and the inhibitory postsynaptic potentials, the phases of action potentials, the role of specific voltage-gated channels, and the concepts of absolute and refractory periods. Here are some questions for you to solve with the answers. Thank you.